let's talk about the quiz for a moment. You have the quiz. The grades are up on Moodle. They're also in your hands. If you have any questions, I'm happy to try to answer them. You know, I graded them real fast, right? I didn't get them until Monday morning, and here we are on Tuesday morning, and they're graded. So all that means is that there's room for human error on my part. So make sure you agree with my grading. If you didn't get your quiz, I can give it to you at the break. So the first quiz is in the history books, right? You have a sense of what you have a sense of what my quizzes are going to be like. This one took you, on average, a half an hour. Some a few minutes less, some a few minutes more. The next one will be longer, so allocate closer to 45 minutes to an hour for the next one. The next one is due on Sunday, so quiz 2 is due on Sunday. And once again, it could cover material in the coloring book, and it could be cumulative, right? Just because you got quizzed on something on quiz 1 doesn't mean you can just forget about it now for quiz 2. Happy to talk about that more in lab, or a break, or now. Will what? The quizzes will always be black and white because I can't print in color. I mean, I can, but I can't. I won't. It's too hard for me. So black and white are going to be the quizzes. So that's your challenge, is to take color information and transform it into black and white information. Plus, you know, three-dimensional information is lost because it's on 2 dimensions. So those are challenging, I do know. All right, today, I will confess that today is probably my least favorite class of the entire year, because we're going to cover a boatload of material, but it's not conceptual. It's taking your concepts, things like ionotropic receptors or G-protein coupled receptors or some chemistry that you know from chemistry. Those are the concepts that I enjoy teaching. Well, I don't teach chemistry, but you know what I mean. And now filling in a bunch of details. And you could say, well, why bother? Why not just why bother with the details? And the point is that the details with things like neurotransmitters and neurotransmitter receptors and the detailed parts of the neuroanatomy, these details are important. It's part of the language of becoming a neuroscientist, which is what we're doing. And so for me to say locus ceruleus, you should eventually, not right now, but eventually know what neurotransmitter I'm talking about and what that means for brain function. That's part of the language. So when I talk about serotonin, you should, things like, okay, I know what serotonin is. It's a tryptophan derivative. I know what parts of the brain it comes from. It comes from the Raffi nucleus. And I know something about the transport mechanisms and the neurotransmitter mechanisms and what it does. So it's detail. It's just detail, but it's important detail for speaking the language of neuroscience, neurophysiology, and neuroanatomy. So I'm gonna go through it, and I confess that I don't love teaching this level of detail, but it's important detail. I'm trying to sell you on the concept that all this stuff matters. So we're gonna talk about classical neurotransmitters. Maybe talk for a few minutes about atypical ones too, because they're more interesting. They're interesting. Neurotransmitter receptors, and we're going to start to talk about neuroanatomy. Okay, so here's a table. I guess we kind of ended the last class with this table. And, tables have a ton of information on them. So this table is partly material that I do want you to know, and partly for reference that I don't expect you to pack into your brain. So now the question is, well, which is which? So let's go through it. Neurotransmitters, the name of the neurotransmitter. Yes? yes, yes, and yes. I do want you to be able to know the names of these neurotransmitters. Acetylcholine is a small molecule that doesn't fall into any of the other categories, so I guess I do want you to know that. Dopamine, norepinephrine, and serotonin are all monoamines. And I do want you to know that. Dopamine and norepinephrine are catecholamines. And I do want you to know that. So you're wondering, am I going to just circle everything on this whole table? I mean, obviously, I hope the answer is no. And I do want you to know that catecholamines are tyrosine derivatives. So both of these catecholamines, that's the definition of a catecholamine, a derivative of the amino acid tyrosine. And I do want you to know that serotonin, even though it's a monoamine, is not a tyrosine derivative, it's a tryptophan derivative. A derivative of the amino acid tryptophan. So it's a monoamine, but it's not a catecholamine. Serotonin. I do want you to know that dopaminergic neurons. So if I talk about a dopaminergic neuron, I'm talking about a neuron, with a cell body and dendrites and axons and presynaptic terminals that releases the neurotransmitter dopamine. A dopaminergic neuron is a neuron that releases dopamine. Not a neuron that receives the dopamine signal. A neurotransmitter that releases dopamine. That's a dopaminergic neuron. The neuron that receives the dopamine signal could be part of a dopaminergic synapse, but that wouldn't be the definition of a dopaminergic neuron. So dopaminergic neurons have cell bodies in an area of the brain called the substantia nigra and the ventral tegmental area. Circle over. Ventral tegmental area. We'll see those anatomically in a few minutes. So when you think about dopamine, I want you to be making these neural connections in your brain. Dopamine, a monoamine that is a catecholamine, which means it's a tyrosine derivative. And cell bodies of dopaminergic neurons are localized in the substantia nigra and the ventral tegmental area. That's a sentence that summarizes what I just circled in orange, at least with regard to dopamine. Norepinephrine, you'd want to say norepinephrinergic neurons. Unfortunately, the language, there's the Atlantic Ocean between England and the United States, and neurons that release norepinephrine are called noradrenergic neurons, because in England, epinephrine is called adrenaline. We sometimes call it adrenaline too, but scientifically we call it norepinephrine. So norepinephrine and noradrenaline, I left out the nor. Norepinephrine and noradrenaline are the same thing. A norepinephrinergic neuron is a noradrenergic neuron. Okay? So, noradrenergic neurons release norepinephrine, which is a monoamine, and it is a catecholamine. And the cell bodies of those neurons are in the locus ceruleus of the brain. We'll see that later. And parallel structure here, serotonergic neurons. Serotonin is a monoamine. It's not a catecholamine because serotonin is a tryptophan derivative. serotonergic neurons their cell bodies are in the Raphe nucleus. Okay, so now, switching gears to the receptors for each of these neurotransmitters. I do want you to know that there are ionotropic receptors for acetylcholine, and there are GPCR receptors for acetylcholine. And I will probably use the language nicotinic for the ionotropic receptors and muscarinic for the GPCR receptors for acetylcholine. The muscarinic receptors come in two categories, but I'm not going to circle or check anything here. I'm not going to ask you to know that the muscarinic receptors are either GQ or GI coupled. But there's a reference point for you if you want to figure out. If you ever need to know, you can look it up on this table. For dopamine, I do want you to know that the receptors for dopamine are all GPCRs. But I'm not going to ask you, and there's subtypes. There's D1 type receptors and D2 type receptors. And D1 type receptors are coupled to GS, and D2 type receptors are coupled to GI, but I'm not circling it. Is it clear what I want you to know and what I don't need you to know? Norepinephrine receptors are also GPCRs. And we have alpha adrenergic receptors and beta adrenergic receptors. They're all GPCRs, different types, decoupled to different G proteins. Let's not worry about the details here. I will, in AMP2, ask you to know this difference, but not here. I do want you to know that serotonergic receptors are of both categories, sort of like acetylcholine. We have ionotropic serotonin receptors, and we have GPCR, many, many, many different subtypes of GPCRs. There were too many even to put into the table. So finishing this out, I want you to know something about function of these systems, the neurotransmitter and the receptor system. So acetylcholine acting on ionotropic receptors is excitatory, just about everywhere. Acetylcholine acting on GPCRs can be excitatory or inhibitory, depending. Dopamine receptors can be stimulatory or inhibitory. There's the letter is right there, so I'm not going to circle that. I am going to ask you to know that dopaminergic receptors are important in several physiological functions. We're going to see them in the motor system, and we're going to talk about them in the reward system, which is related to addiction. I'm not going to ask you to know right now that abnormalities in this pathway are associated with some disease states. So we're not going to talk about pathophysiology too much, although it's really interesting. We're going to talk mostly about physiology because that's the name of the course. And ionotropic receptors, GPCRs, dopaminergic GPCRs are important in these physiological properties. addiction is not really physiology, so in principle we could leave addiction out. But reward is physiological. You do things that are rewarding. Sleeping, eating, drinking. You get a reward signal when you drink when you're thirsty. That's how you know to drink. Norepinephrine, I'm going to ask you to know that it's associated with general arousal. Being alert, being awake, being attentive. And serotonergic receptors are associated with general mood. And so abnormalities of mood, depression, mania, in a bipolar situation, are associated in many cases with abnormalities in the serotonin pathway. Questions about what I want you to know about these major classical neurotransmitters. I want to diverge for just a moment and ask you, so what do you think are and some of you may know this from another course, what are the definitions of a neurotransmitter? What makes a chemical a neurotransmitter? We've got lots of chemicals in our body, but not all of them are neurotransmitters. Sophie?
It originates from a neuron?
Yeah, neurons make them. If neurons don't make the chemical, it's not a neurotransmitter. So neurons make the chemical. What else is another factor that defines a neurotransmitter? There's a clue on the board. So is it enough to say that the neuron makes the chemical? What does the neuron have to do with that chemical after it makes it? Target it? Yeah, it has to put it somewhere, and then Charlie?
It has to be released.
It has to be released at the synapse.
Membrane through the synaptic cleft of the postsynaptic.
Right. So the chemical has to be packaged into presynaptic terminals and released into the synaptic cleft in response to some stimulus. So synthesis, packaging, release. What else do we need? It's on the board. Is it receptors? It has to have something that receives that signal. Those are pretty much the core definitions of a molecule that is defined as a neurotransmitter. It's going to get fuzzy when we get into neuromodulators, but those are the core definitions of a neurotransmitter. So, any questions about those before I go on? Okay, while I'm erasing, those of you in the far edge of the room, over up here on this side, can you see when I write over here? Is it too far away to see? Are we getting all kinds of glare on this one?
Up in the right-hand corner.
In the
but
upper right-hand corner, it's glary? Okay. Not sure I can do anything about it. Okay. So now, oops. Let's go to some more neurotransmitters. So these are more classical neurotransmitters. I want you to know that glutamate is an amino acid. You already know that, but it's also a neurotransmitter. I want you to know that glutamate receptors can be either ionotropic or G protein coupled receptors. and we call these metabotropic. Metabotropic. So ionotropic receptors are ligand-gated ion channels. Metabotropic receptors are GPCRs. So you could go back to muscarinic receptors and say, are muscarinic receptors metabotropic receptors? And the answer is yes. But the M's get confusing, so that's why I put this new M word here. Muscarinic receptors are metabotropic receptors for acetylcholine. And I want you to know that these are generally excitatory. So glutamate is the most important excitatory neurotransmitter in the central nervous system. And we'll talk about we'll talk about some types of ionotropic receptors, and we'll talk about their function later. Not circling here. GABA, you've already heard of GABA from the problem set. It is an amino acid, but it's not one of the amino acids we build into proteins. There's thousands of amino acids, and only 20 of them are used in proteins. There are ionotropic GABA receptors and metabotropic GABA receptors. And they are both inhibitory, but GABA A receptors are ionotropic GABA receptors are inhibitory because of chloride channels, and metabotropic GABA receptors are inhibitory because of their coupling to potassium channels. You did this in the problem set. I do want you to know that glycine is an important inhibitory. You already know that glycine is an amino acid, so I can circle this without feeling like I'm adding to your cognitive load. And let's just say that glycine are another set of inhibitory. Glycine receptors are another set of inhibitory receptors. And there are purines. ATP, it turns out, is a neurotransmitter. So in addition to all that good stuff that ATP does to all your cells, it's a neurotransmitter. And its breakdown product, adenosine, is a neurotransmitter. And we have different types of receptors for them, and they have multiple functions. And you can see I'm not circling anything, because I'm not going to test you on purinergic receptors. But they're important neurotransmitters. And important neurotransmitter receptors. Those of you who are caffeinated right now are having the effect of caffeine because caffeine inhibits a subtype of adenosine receptors. So it's important in pharmacology. Questions about this set of information? Remember, what I'm trying to do is not overload your brain, although I know I am. What I'm trying to do is speak the language so that we have common language. So when I talk about a GABAergic neuron, you're already thinking, okay, inhibitory, is the receptor an ionotropic or a metabotropic receptor? But I know that GABA's got to be inhibitory. Let's start there. When I talk about a glutamatergic system, you should be thinking, okay, excitatory. Somehow excitatory, either ionotropic or metabotropic, but it's going to be excitatory pathway. Questions? So now I'm just going to go through them sort of one at a time with a little bit more detail. I gave you all these slides, so let's figure out what I want you to know from this. Here's the structure of acetylcholine, but I'm not going to ask you to know it. This acetate choline. Acetylcholine. Small molecule. Molecular weight of, I don't know, 100 or something. One thing that I do want you to know about acetylcholinergic neurotransmission is that unlike most of the other neurotransmitters that we talk about, acetylcholine is hydrolyzed by an enzyme in the synaptic cleft. So its removal has an extra step from other things. It is hydrolyzed, it's broken down. It's broken down into acetate and choline. And choline, the choline piece, is reuptaked by a sodium-dependent syn porter, like we talked about last week, generically, about how neurotransmitter is retaken up by a neuron from the synaptic cleft. Remember, there's that sodium neurotransmitter cotransporter, and then the proton neurotransmitter antiporter in the synaptic vesicle, right? I am going to ask you to know those concepts. But in this, for acetylcholine, choline, not acetylcholine, is what is taken back up and then resynthesized using choline that we just took back up, and acetyl-CoA, something you're familiar with from cell physiology. Okay, so that's the detail that I want you to know from this information about acetylcholine. I don't think there's any, Sophie.
So just to clarify, you said that the NA-choline transporter is a syn porter, or?
The sodium choline cotransporter in the surface membrane of the presynaptic terminal that is responsible for taking choline back up from the synaptic cleft after it's been hydrolyzed is a syn porter.
And why would the, oh, no.
I think everything on this is a slide for visual interest, because everything that I mentioned on the table is re-mentioned here. We've got nicotinic receptors, which are ionotropic. We've got muscarinic receptors that are metabotropic. This is all acetylcholine. I'm not going to ask you to know the subtypes of muscarinic or nicotinic receptors. It turns out that there's probably 3 major subtypes of ionotropic, and you can see 5 different subtypes of metabotropic. You go into advanced neuroscience, neurobiology, neurophysiology, this becomes important, but not to. You should be able, I mean, I hope you can tell. Which type of receptor is this cartoon? Point over here. Which type of receptor? Is this a ribbon diagram? Oh. Anyway. Is it metabotropic or ionotropic? Nope, that's a metabotropic receptor. How do I know? Because that's an ionotropic receptor. If you don't see a pore, it's not a, it's a metabotropic. Oh, and two ways to know. 1, 2, 3, 4, 5, 6, 7 transmembrane. The membrane is here. A bundle of 7 alpha helices is a defining characteristic of all metabotropic receptors, all GPCRs. Having a pore is the defining characteristic of an ionotropic receptor. Okay, questions about acetylcholine, because we're going to move on. Okay, so now we're going to talk about catecholamine. Well, we're going to talk about monoamines. A subtype of monoamine are the catecholamines. This ring structure defines a catecholamine. So I'm not going to ask you to know the structures of dopamine or norepinephrine or epinephrine, but I am going to ask you to be able to recognize this structure as defining a catecholamine. Catecholamine. This is the catechol group, and this is an amine. Chemistry. Your favorite. So, catecholamines have this general structure. Dopamine is a catecholamine. You already know that. I've mentioned the substantia nigra. I've mentioned the VTA. I've mentioned that it's important for motor control and for reward. So this is just reinforcing, letting me use the language again and again. Let me just circle two things, though, that are on this slide. So the substantia nigra is the location of the cell bodies of dopaminergic neurons. The axons of those cell bodies go to the striatum, and let's just talk about the striatum. We'll see the striatum when we get into the brain. So, substantia nigra is where the cell bodies are, and the striatum is where the terminals, the synapses, from those neurons are. Okay, that's the target. Similarly, the cell bodies of these dopaminergic neurons in the ventral tegmental area project to the nucleus accumbens. So N almost always is going to become nucleus. Nucleus accumbens, which is another part of the brain, and that's where the synapses are. And this pathway from the substantia nigra to the striatum is important in motor control, and this pathway from the VTA to the nucleus accumbens is important for reward in addiction. That's information that I could expect you to understand or tell me about. Questions about dopamine? And here's the structure of dopamine. It's a catecholamine. The dopaminergic pathways, their cell bodies are in the substantia nigra and VTA. The substantia nigra and the VTA are in a part of the brain called the midbrain. So I might as well start using some neuroanatomy. Midbrain. That's the midbrain. So the substantia nigra and the VTA are part of the midbrain. These lines are showing where the axons go. These are the axons to the striatum, and it's not very well shown, but the nucleus accumbens is somewhere around here, so maybe let's just draw another arrow here to the nucleus accumbens. Okay, so this is where the cell bodies are, and the lines show arrows of axons, and you can see I'm asking you to know only a subset of them, to the striatum and to the nucleus accumbens. And that's where the synapses are. I don't want you to know any chemistry of the catecholamines, except for the fact that you should, like I said, you should be able to recognize them. So here is tyrosine, but here is the catecholamine. So dopamine is a catecholamine, norepinephrine is a catecholamine, and epinephrine is a catecholamine. One bit of information that will help you in the future is knowing that all of these pathways start with an enzyme called tyrosine hydroxylase. And it's the rate-limiting step for the synthesis of all of these neurotransmitters. So tyrosine is the source amino acid. Tyrosine hydroxylase is the enzyme that defines whether a neuron is either dopamine, is catecholaminergic. Because all catecholaminergic neurons have to have a pretty high expression of this enzyme in order to make their neurotransmitter. That's pretty much the main information that this slide embeds. I don't want you to know the structures. I don't expect you to know that dopamine comes first and then norepinephrine and then epinephrine. I don't need you to know that. I don't need you to know the names of any of the other enzymes, but it's there on your sheet for reference. But I do want you to know the name tyrosine hydroxylase. Because it defines catecholaminergic neurons. Dopamine receptors are all GPCRs. 1, 2, 3, 4, 5, 6, 7 helices. Expressed widely. Some are excitatory, some are inhibitory. Valentine's Day is coming up, so I put this in here. When monogamous mammals find a mate, which is a rewarding event, there is a higher expression of the excitatory dopaminergic neurons in their nucleus accumbens, and that leads to a boost of the reward pattern. So there are some types of voles that mate for life. Some birds mate for life. Human beings, it's a mixed bag. This vole is getting rewarded. Okay. Interesting side effects or side information about dopamine receptors. Dopamine receptors, I don't know why I put this slide up again. It's the same slide. Dopamine receptors, their cell bodies are in the ventral tegmental area and the nucleus accumbens, and they project to the striatum, motor, we'll get to that, and ventral tegmental area, which we just talked about. So this is just a review. Switching gears to another catecholamine, norepinephrine. The cell bodies of noradrenergic neurons are localized in a part of the midbrain, again, called the locus ceruleus. Oh, by the way, let me go back. Those of you who know Latin might know that substantia nigra means black substance, because it's dark. It's in the brain, we'll see it, maybe, I hope, as a dark spot in the midbrain. Anybody know enough Latin to know what locus ceruleus means? What do you suppose locus means? Location. What is cerulean? It's a blue. So the blue location. It's bluish in color, I guess, in fresh tissues. I've never seen it look blue because I've always only looked at fixed, embalmed tissue. Okay, anyway. The locus ceruleus is the location of the cell bodies of noradrenergic neurons, and the axons go everywhere, because as you can expect, something that is important for arousal is going to change the brain all over the place. So we don't have to talk about specific targets, because it goes everywhere. And we talk about sympathetic nervous system in the other AMP class. So even though the other AMP class does not focus on the nervous system the way you guys are, this becomes part of cardiovascular, respiratory, and renal physiology. But the point is, well, let me just leave it like that. Adrenergic receptors are all GPCRs. We've got subtypes. That's all this is just a reference slide from future reference. If you want to know, okay, I'm reading a paper and it's talking about beta-1 adrenergic receptors, you should know that it's coupled to GS, but I'm not circling it because I don't want you to memorize that for this class, but those of you who've taken AMP2 know that that is a coupling of information that we do need to know in AMP2. Questions about adrenergic receptors? Serotonin is a monoamine, but is it a catecholamine? No, it doesn't have the catechol group. It's not a derivative of tyrosine. It's a derivative of tryptophan. I don't expect you to know the structure, but I do expect you to be able to say, that's a monoamine, it's got one amino group, but it's not a catecholamine. Because if you know what a catechol looks like, you can say, oh, it doesn't have it. The cell bodies of serotonergic neurons are in a part of the midbrain, again, called the Raphe nuclei. And they go everywhere. Because something like mood is a very complicated phenomenon, and so those neurons go to lots of different locations. By the way, here's an important bit of information. Serotonin is also called 5-hydroxytryptamine. And it's abbreviated 5-HT. So I'm going to use the word serotonin. But I just want to point out that if you go into the literature and you see 5-HT, the 5-HT1 receptor, for example, it's a serotonergic receptor. It's a serotonin receptor, because 5-HT is serotonin. So we don't say serotonin 1 receptors and serotonin 3A receptors. We talk about 5-HT3 receptors and 5-HT7 receptors. I will use the word serotonin. And if I make a mistake and I say 5-HT, just catch me up. Say, we're talking about serotonin, right? Oh, yeah, that's right. And so like I said, there are many 5-HT receptors. Some of them are inotropic. Most of them are metabotropic. Here's a metabotropic serotonin receptor. And we've already talked about how serotonin, so what is this? This is a mistake. This slide has a mistake on it. This arrow should be going this way. This is a sodium serotonin co-transporter. To fix that, this is a sodium serotonin co-transporter.
Did you switch the direction of the arrow?
I did. I switched the direction of the arrow. That's what I did, because this arrow is wrong. It's not an antiporter, it's a co-transporter. Somebody screwed up. And it wasn't me. But I made the mistake of choosing this figure. Because it's nice and simple, right? Serotonin is released by the presynaptic terminal. There are 5-HT receptors in the postsynaptic terminal. Here's the synaptic cleft. Serotonin is retaken up in a sodium dependent way. I almost drew on the screen. Sodium dependent co-transporter, and then a proton dependent exchanger to package serotonin back up into vesicles, right? Why am I doing this again? Because this protein is the target of a class of drugs you've heard of, the SSRI class of antidepressant drugs. These drugs inhibit the serotonin transporter. And that's why they're important in mood disorder, antidepressant. Okay, there are lots of other neurotransmitters. We could talk about nitric oxide is a neurotransmitter. Carbon monoxide is a neurotransmitter, but weird enough. There's endocannabinoids, so the thing that is activated by cannabis. Endocannabinoids are neurotransmitters. Endo-opioids are neurotransmitters. We have receptors for opioids. That's why opiates work. What am I going to ask you to know? I'm not going to ask you. Oh yeah, I'm going to ask you to know the sequence of this, right? And I'm just not going to do that. All I want you to know is that there are peptide neurotransmitters. Opioids are peptides. And so when we talk about pain, we'll talk about opioid modulation of pain. So let's take a break. We're going to transition from this wide-ranging discussion of classical neurotransmitters to a wide-ranging discussion of neuroanatomy. So again, just trying to get the language of neuroanatomy out there. And the answer key to the quiz is up on Moodle. So you can check your knowledge and you can check your answers, the one that you turned in, to see that what you missed. All right, let's do some neuroanatomy. So my goal is to just use some of the language of neuroanatomy. You are going to have to, it's a lot of nouns. Teaching it is particularly uninspiring because it's just a lot of nouns. It's not very conceptual. But let's do what we can do. So here's a cartoon from your textbook that colorizes the different parts of the brain. We're going to focus on brain neuroanatomy. So we have different lobes: frontal lobe, parietal lobe, occipital lobe, and temporal lobe. This is a sagittal superficial view. looking at the brain from the side. Here is a midline sagittal section, so this is cutting the brain. Frontal lobe, parietal lobe, occipital lobe. Can't see the temporal lobe. Oh, yeah. Is that what you meant? So the lobes of the brain are important to know. Cerebellum is another, it's not a lobe of the cerebrum, but it's another easy to identify part of the brain. There's a whole other set of words that I don't use very much. The cerebrum is called the telencephalon. This slide called, in here, down in the sort of the core, inside the brain, is the thalamus, which is also known as the diencephalon. And the midbrain is called the mesencephalon. So there's this encephalon language. Telencephalon, the cerebrum. Diencephalon, pretty much synonymous with the thalamus and hypothalamus. Mesencephalon, pretty much synonymous with the midbrain, with the brainstem. So, but I don't use that language, but you'll come across it. So this slide is something, all the words on this slide, except for diencephalon, could be on a quiz. So you could cross off diencephalon and realize everything else is something to know. So, here's a picture you've never seen before, a set of pictures. And we're going to do a challenge game here. Some of you know some of the things here. So let's just get oriented. Here is a superficial side view. Here is a superficial superior view. And if I showed you this on a quiz or on something else, I would have to get you oriented. I think you could tell that that was a superior view, and you could tell that it was superficial superior view. But I'd have to tell you, this is the front, this is the anterior, and this is the posterior part of the brain, because that's only fair. I would tell you that. And I would say that this is the left and this is the right side. Or maybe not. But. By the way, I took off a point. If you circle, I think on the quiz, just to be clear why I took off that point. When I asked you to point to a distal part of the left arm, I meant this person's left arm. And many of you went to the right, this person's right arm, which isn't correct. The right and the left are not the same. So you have to, one of the things to do is to know, okay, wait, and this person's left arm is on your right, because you're looking at it. So one of the first things I do with anatomy is I put up my orientation. This is left, this is right. No, on that piece of paper, this is left and this is right. Right? Just so you know. Okay, so let's play this game. Easton, I'm going to pick on you because you're in the far corner. Here's the laser. You're going to point to something on that screen that you know the name of. Some of it starts easy and it gets harder, and you're going to label it. Now I'm going to raise this. I'm going to raise this screen again because I'm going to write on it. So you can point to either screen and give me a name.
That would be cerebellum.
That is the cerebellum. And just to be clear, so is this. Okay? Joey.
The corpus callosum.
The corpus callosum.
And then, which way do you want me to go? Do you want me to go across
Go
the back?
The corpus callosum are myelinated axons that connect the two hemispheres of the brain. In fact, I just gave you a word that you could use to label. Temporal lobe. Okay? In fact, this is a little bit of the temporal lobe right there, sort of behind what we're seeing. Okay, Ashley? Okay. Brainstem. Okay. Neil?
There's the frontal lobe.
Yeah, frontal lobe. Charlie? I can come over here.
Okay,
I
I'm
just
just trying to
can't
make
draw.
sure.
Well, maybe I'm gonna need to know more. So we'll see how this works.
This one, the parietal lobe would be like somewhere over here.
Yeah. I'll continue over here, but then this gets too complicated. So the parietal lobe, something like that. Okay. Danielle? Pons. Show me, let's make sure you're accurate. T is the pons. So this area right here is the pons. See, some of you know this stuff already. Sheldon. Good, the thalamus.
Just like this whole half, even
A hemisphere. Show me it, actually, show me the right hemisphere. Okay, so this is anterior, this is posterior, this is left, and this is right. So this is the right hemisphere, the right cerebral hemisphere. That's a legitimate label.
Is this the medulla oblongata?
Good, okay, right here, this part of the brainstem is the medulla oblongata. You will want to know that name. You want to play it?
Yeah.
Okay, Eric?
Midbrain.
Okay. The midbrain. Another part of the brainstem is called the midbrain. This is where the locus ceruleus is, the substantia nigra, and the, what's the third one? Raphe nuclei are in the midbrain.
The occipital lobe.
Occipital lobe. Okay, so I'm going to stay over here. Grab a chair. No, I'm going to do it over here. All the way over. Occipital. Also over here. Occipital. Okay. Chelsea, see, it's getting harder. The easy ones are being picked. So now it's getting harder. Give me something.
Would the hypothalamus be part of the midbrain?
Close, but not quite.
Okay, so is he like right under the thalamus then?
Hypothalamus, so you're translating the word very, very good. Hypothalamus is here. So it's hypo and anterior to the thalamus. This is the hypothalamus. We're going to see that big time when we get to endocrinology. Okay, Tyner?
Central sulcus.
Okay, so we got a ringer. So let's put up the word, so what is a sulcus? Just generally. A groove, a fissure. So this right here, and this right here. Those are, they probably have a name, but I don't know what they are. Those are two, each of them is a sulcus. The two of them are sulci. Latin. So, what Tynan knows is that this sulcus right here that separates the frontal lobe from the parietal lobe has a special name called the central sulcus. It is a sulcus, and it has a specific name, the central sulcus. Matt?
I think the substantia nigra would be O.
Oh, yeah, I guess that's true. O is the substantia nigra.
Somewhere around right here is the cerebral cortex.
Okay, yeah, we have, I mean, this whole thing,
All of it is.
this whole thing is the cerebral cortex. And it happens to be the right hemisphere. So the right hemisphere of the cerebral cortex, because you can tell this is the front and this is the back. So this is the right side. So this whole thing is cerebral cortex, not just one spot.
Okay.
Okay, so yes. So the hypothalamus is connected to the pituitary gland, so E is probably the pituitary. We're going to see that big time when we get to endocrinology. Okay? Rita? Nothing? Okay, pass it back. Okay, Ruthie, got anything for me? Say again, basal ganglia.
I might be wrong.
You're very close. The basal ganglia are there, but we can't see them in any of these sections. Sophie, you got anything for me? So I'll start pointing things out and asking if anyone knows the name of it.
The longitudinal.
Yeah, point to it.
sulcus? This thing?
Yes. This thing that separates the two hemispheres has a name called the longitudinal fissure, but it's a sulcus. Longitudinal fissure. Okay, so I want to use some. Now I'm going to use some new. I'm going to point out some things, and I'm going to ask you to anybody, to come up with the name. Okay, this is the longitudinal fissure. What is this sulcus right here called? The sulcus that separates the temporal lobe from the frontal and parietal lobes. So let me use the language. The central sulcus separates the frontal lobe from the parietal lobe. X, that you're naming, separates the temporal lobe from these other two lobes. What is the name of X?
The horizontal sulcus.
Say again?
Sulcus lateral
The
something.
lateral sulcus. Very good. The lateral sulcus or the lateral fissure. Did you know that, or did you just pull that name out? You just thought it might be logical?
Logical.
Okay, good. Good logic. So this is the lateral sulcus or the lateral fissure. Okay, let me go over to the other side. If I go to the other side of the screen, can you folks see it? Even down here? Okay, this is the central, actually, I probably did it wrong. This is the central sulcus. So obviously I will take some, I did it wrong, so. This is the central sulcus. What is the name of this structure of the frontal lobe? Anybody? Sophie?
Isn't it like the somatosensory?
Close. That's the function, but it's got a name. It's got an anatomical name. It's not the somatosensory cortex. But you're close. You'll see in a moment. So this is the central sulcus. What is the name of the lumps? So here's a sulcus, and here's a sulcus, and there's this lump of brain between this groove and this groove. What's the name of those things that we call those lumps? I don't call them lumps, we call them a gyrus. So this is a gyrus. The plural is gyri. So I'm going to give you a hint. I'm going to ask you to use your sort of pattern recognition features of your amazing human brain. This thing is called the superior temporal gyrus. Superior, above, it's high in the temporal lobe, so it's the superior temporal gyrus. This is the central sulcus. So what's the name of this structure right here?
Posterior frontal gyrus.
That would be a logical name, but it's called the pre-central gyrus. Pre, before. So this is the pre-central gyrus. And what do you, now you can tell me, what's the name of this gyrus back here? Post-central gyrus. Okay, so some of the names make sense to you. You know one, you know two. Pre-central gyrus, post-central gyrus. Those are anatomical names. Sophie used a functional name. She said the somatosensory cortex. Somatosensory, soma, body, sensory, sensing our body. The part of our brain that is dominated, that does sensation of our body, is in the post-central gyrus. This is also known as the somatosensory cortex, or the somatosensory gyrus. So this is an anatomical name, and somatosensory is a functional name for the same thing. Somatosensory cortex, or somatosensory gyrus.
That's the post-central.
That's the post-central. The pre-central gyrus has a function. It's the motor cortex. It's important for movement. So you could label the pre-central gyrus with two names: motor cortex, pre-central gyrus. Okay, this is fun. Okay. anyone know the name, the functional name for this part of the occipital lobe? The occipital lobe, the very back of it, has a function, a specific function. Does anyone know the name of that functional part of the brain?
See.
That's the visual cortex. And I'll do one more, and then I'm just reminded of something else that I said I would do. This part of the superior temporal lobe is the auditory cortex. So vision, hearing in the brain. We're going to pick this up on Thursday, but this is your challenge is to learn it. We've got a visitor here who wants to talk to us about
The Philadelphia Center.
the Philadelphia Center. So please stay for 5 minutes to hear what, I'm forgetting your name.
That's all right, Morgan.
Morgan is going to tell us about the Philadelphia Center.
Thank you. Thank you so much for letting me interrupt your class time. Thanks for having me. Hi, everyone. My name's Morgan Madavi. I'm the recruiter with the Philadelphia Center. I'm also an alum of the program. I did the program in 2011 when I was a sophomore at Kalamazoo College, so I'm excited to be here to share about the program with you all. It's been really instrumental in my life as a person, as a professional, and as an academic since 2011. Have any of you heard of the program before? Yeah, have you heard me speak in your classes before? All right, well, apologies that you get to hear the same presentation over again, but I'll be quick. So the Philadelphia Center, we're an off-campus program based in Philadelphia that offers you the opportunity to see what it's like to move to, live in, and work in a new city. We run programs every fall semester, spring semester, and we run an 8-week summer program. I'm going to give you kind of an overview of the semester program, and then I'll share a little bit about the summer program specifics. As I said, you move to the city. In that first week, we put you up in a hotel right around the corner from our office in Center City, Philadelphia. And the first week is our orientation to the city, and is really focused on you finding independent housing. So we set up apartment viewings for you. You can go on Craigslist. You determine who you're going to live with among the participants of the program. Get to know the different neighborhoods of Philadelphia. We orient you to SEPTA, our public transportation system. Within those first 7 days, you sign a lease and move into an apartment. Which sounds really fast, but we've run the program for 52 years. No one's ever gone homeless. We guarantee you will find housing. As you're looking for housing, you also sign up for classes. Everybody takes one seminar course and one elective course taught by our Philadelphia Center faculty. And our whole program, and the classes in particular, are taught in the pedagogy of experiential education, which is a style of teaching and learning that's built out of the idea that learning happens everywhere. So the classes are very hands-on. You're learning in the classroom, you're getting history, background, theory, but then also going out into the city and seeing things in practice, which is a really fun and different way to learn. It really gets you to know the city of Philly in kind of a different way. That was one of my favorite components of the program, actually. From your seminar course, you get your faculty advisor, who works really closely with you through all aspects of the program. So you work with your faculty advisor then to find an internship. We have over 850 internship placements in kind of anything that you can imagine, every major professional field. So you work with your faculty advisor to think about, what are your goals for your time in Philadelphia? How does it connect to your goals back here at Earlham? And then how does it connect to your goals after graduation 5-10 years down the road? What kind of work environment do you want? What kind of supervision? What would you like to be doing? Then you work on your resume and your cover letter to refine them to each of the positions where you'll be applying. And we have everybody interview at least 3 placements to get that practice with professional interviewing. And then once you interview, you determine where you'd like to work, and then you intern full-time, 4 days a week for the whole semester. So that's kind of the overview of the program, the components being independent city living, academic and intellectual growth, and professional development through the internship. I mentioned we have over 850 internships, couldn't tell you them all even if I tried, but we have lots of different things that you can do. Can you all tell me some of the majors that we have in the room here?
This is mostly neuroscience,
Mostly neuroscience?
biology, biochemistry.
Okay,
I
great.
think that covers
Okay,
most of it.
so we got a lot of placements. So we have a placement with the University of Pennsylvania, and they're doing research in the neuropsychiatry unit. We also have a placement at Thomas Jefferson Hospital working in neurology research. We have lots of placements at University of Pennsylvania doing biology, working as a biology research assistant, as well as some other research placements like at the Children's Hospital doing pediatric psychology research. Those are just some examples. Then we also have some, direct service placements, whether that's working in Thomas Jefferson Hospital in their emergency room, or working in For example, the Defenders Association is in the Juvenile Special Defense Unit as a legal and social work intern. We have placements working with in the Prison Society or working with Nationality Service Center, which is a organization that works with refugees and immigrants, doing psychological support, and then also some community-based support. So lots of different opportunities there. I can share more specifics about specific internships if you're interested. But suffice to say that all of our internships, so our whole program, you get a full semester's worth of credit, and because of that, we ensure that the internships are really about your learning. So you don't need to have any experience to get an internship. It's really just about showing you what is it like to work in this field. And you're not going to be tucked in a corner making copies or getting coffee. You're really doing hands-on work, seeing what it's like to learn in the field. We have a vetting process for our supervisors to ensure that they're there for you, they're invested in your learning. So you're going to get a lot of support, a lot of growth, really see what it's like in the to be in that field. We see a lot of our alumni get job offers through their internship placements, whether that's at the site where they interned, or there's a lot of networking built into the semester. So your supervisor will likely introduce you to lots of different people. We also have lots of alumni. We have over 9,000 alumni, a lot of whom are in Philadelphia. So there's opportunities to meet with them. Last semester we had a student who connected with an alumni. He went back to his home campus and then emailed the alumni, and the alumni got him a job. So there's the ways that the sort of professional impact of this really ripples out for folks. I think that's what I'll say about the internships. I talked a little about the classes, and then the last piece is Philadelphia. Have any of you been to Philly? Yeah? Okay. Philly's a great city. People kind of forget about Philly. It's the 5th largest city in the U.S. We're an hour and a half from New York. You can get bus tickets for $10, 2.5 hours from D.C., an hour from Baltimore. Philly itself is great. There's a whole neighborhood called University City. At any time, there's something like 300,000 college students, so it's a young city. With your student ID, you can do something for free every night of the week. Go to museums, go to sports games, go to shows. Eat different food. It's a food city. You can eat food from around the world in like a 10-block radius. And that's part of the program, is what is it like to move to a new city? How do you get to know your community? Some of the adulting things like, grocery shopping, cooking for yourself, time management, all of that. But also, trying out different activities. What is it like how do you meet people when you're not put in a dorm with, hundreds of people your own age? It's kind of hard to meet people after college, turns out. So you get some of those skills that are very helpful and important. So yeah, I'm happy to share more about Philly. I love it. I'd never been before doing the program and moved back even before I got this job, just because it's a great city. It's easy to navigate to. I'm directionally challenged, and I was able to figure it out in a semester, so you all got this. A couple of different logistical things. If you are interested in applying for the fall, we are still accepting applications. The first step is to meet with Jennifer Lewis in the Center for Global Education. We're also accepting applications for the summer. They're due March 16th, and you can just apply through our website. The summer program is a similar structure, 8 weeks rather than 16. You work a full-time internship, you take 1 class rather than 2, and then, what is the other piece? Oh, you live in suite-style apartments at the University of Pennsylvania, because 8 weeks is just too short to get a lease. In terms of cost, you pay Earlham, for the semester program, you pay Earlham as you would normally. All of your scholarship, financial aid, everything transfers. However, because you're not living in the dorm, Earlham will then cut you a check when you arrive in Philadelphia that's something like $5,500 to cover your living expenses while you're in Philadelphia. So that's kind of nice. And you get to budget and find your apartment, so some students actually do end up saving money, and then all that money, the rest of it stays in the bank account. So you can work that out as works for you. For the summer, there is a fee associated. You get it for the semester, full semester's worth of credit, move you along towards graduation. The summer also you get credit, 8 credits rather than 16. So it was a lot really quickly. I've had a lot of coffee and I'm from the East Coast, so I hope you understood some of that. I will be tabling in outside the Runyon Center between 11 and 1 if you'd like to come chat with me. I'm also doing an info session at 4 in LBC 124. There'll also be some Earlham alum to share more insight as to what was in the program. Thank you all. Thank you for having me.
A lot of my advisees have done the Philadelphia Center. It's been life-changing. It's really been very pivotal in their long-term growth. And I will also testify, Philadelphia is a great city. My daughter lives there. We visit frequently now that I'm a grandfather. And so Philadelphia is a great city. So it is a program worth really considering.